And with that, I would like to introduce our guest speaker, Professor Pam Ronald. Pam graduated from Reed in 1982, and I see a few of you from the class of 1982 joining us this evening, which is great. And she majored in biology, that's fitting, and is currently a distinguished professor in the Department of Plant Pathology and the Genome Center at UC Davis. She's recognized for her research that contributed to the development of high-yielding rice varieties grown by more than 6 million subsistence farmers in India and Bangladesh. She was named one of the world's most influential scientific minds by Thomson Reuters and one of the world's 100 most influential people in biotech by Scientific American. She's also a member of the National Academy of Sciences. Pam, thank you so much for being here, and I'll hand it to you.
Thank you, Olga. Thank you very much for putting this talk together. And it's great to be here. And I'm very excited to see some of my old Reed buddies. Hi, Granger. Hi, Rebecca. I hope we have a chance to talk at the end. So first, let me ask Olga if you can see my slides. I think so.
I don't see your slides yet.
Ah.
Oh, yes, I do. They're in there.
Okay, good. Well, let's get started then. So I know there's quite a spread of ages and some freshmen and some people that have graduated from college many, many years ago. So I thought I'd just give a brief overview of how I started my scientific life. And it was here in Desolation Wilderness in the Sierra Nevada, where my brothers and I went backpacking and learned about the flowers, and we lived up there in the summers. Now, after that time, I went to Reed College, and I heard there were a lot of interesting people and that you could study biology, and I found this great old picture of our biology professors at that time, and I wanted to just call out a few people. So Bert Brehm was really a fantastic botanist, and he allowed me to learn a heck of a lot more about flowers, and I began to feel even more comfortable in the wilderness knowing the names of so many flowers and their relationships, and that's been really an important part of my life ever since I was at Reed. Peter Russell, some of you may know him, he's just a fantastic geneticist. He really made genetics exciting, and I didn't know any scientific researchers, and he was really the first person that introduced me to the idea that biology is all about discovery. And of course, genetics has a central role in all of biology. And he also was a great writer. He wrote these really fantastic genetics textbooks. There's Lawrence Rubens, who I worked in his lab in the summers, and I'm not really sure why he hired me, except he's a super nice guy, but I was a little squeamish around the frogs. And but he was very influential. Not only him and Judith brought me into their home and fed me lots of wonderful meals, but I realized I really couldn't work with animals, and I quickly shifted to plants that don't bleed. Now I wanted to, special thanks to Helen Stafford, who was featured on the cover of Reed magazine a number of years ago. She was the first female professor in all of sciences at Reed, and really dedicated to her work. And she was a little skeptical of us vegetarians. I remember going to her house, and she prepared food for us and prepared vegetarian food, but she was a little grudging about it. She said, plants have feelings too. Now, she also introduced me to this idea that plants and microbes talk to each other. And it was really stunning to me. I never really thought about how plants and microbes form associations and why some plants are resistant to microbes and some get susceptible. And just to give you an example, this rhizobium is a bacterium that forms a symbiotic relationship with plant roots and helps the plant fix nitrogen from the atmosphere. So it's very, very important for agriculture. And it's a very sort of intimate molecular dialogue between these two species. And here's another example of a parasite. So this is a fungus called, actually it's a plant parasite called striga that actually parasitizes the root of its host. And it's a huge, huge problem for farmers in Africa. And then importantly, she told me about mycorrhizal fungi. So these are fungi that form symbiotic associations with many different plant species. And here is shown a pine seedling. And I'm showing you that because I got very intrigued with this idea that the fungus forms like this extra root system and helps bring nutrients into the plant. And then very conveniently, 40 years ago now, Mount St. Helens exploded and everything was wiped out. And I was able to meet some researchers that were studying recolonization of Mount St. Helens. And it was very fun for me because I like to go on adventures, and we would head up to what was called the red zone and collect soil samples and try to see if there were mycorrhizal fungi that could survive the explosion. And so that led to my senior thesis, and it also led to my understanding that control experiments are really very, very important, and you can't really make firm conclusions if your controls don't work out very well. But I certainly enjoyed that experience. Okay, so I'm mentioning these symbiotic and these plant microbe communications because I have spent my entire last 40 years working on this, this biological problem. And it might seem a little strange to spend so much time on this, but I hope by the end of the talk you can understand how very exciting this is. So just a little bit of history. In 1845, the late Blight destroyed the Irish potato crop. And some of you have probably heard of the Irish potato famine, where a million people died, 1 million more left Ireland, came to the United States and changed world history. And one of those immigrants was Barack Obama's great-great-grandfather. Now, at the time, no one knew what caused this serious infection, and it was thought to be an act of God. And it wasn't until 1853 that Anton de Berry demonstrated that their microorganisms actually cause disease on cereals. And he was looking at these rust and smut fungi. And then in 1862, Louis Pasteur showed that bacterial contamination caused diseases, what he called diseases of beer and wine. And it wasn't until 1876 that George Koch showed that animal diseases are also caused by microorganisms, and he was studying anthrax infecting mouse. And this is very significant for plant biologists, that plant biologists were the ones that discovered the microbial basis of disease. Okay, so how do plants recognize microbes? So these early studies launched this very interesting time of resistance breeding. And it was in 1905 that Roland Biffin, who was a British geneticist, figured out that there were some resistant wheat varieties and some susceptible wheat varieties, and that you could actually cross-pollinate those wheat varieties, and the progeny, the next generation, would be resistant to infection. So this really launched the introduction of resistance genes into many different plant species. And so today, virtually everything we eat carries genes for resistance, and they're very important for economic and environmental aspects of farming, because you don't need to spray anything if the plant's resistant, and it also is much more economically efficient for the farmer. So despite these early studies, when I started my graduate work, no one knew what these genes were. So we've been eating these genes for many, many years. No one knew what the genes were, but there were starting to be some very interesting molecular tools. And so I got drawn sort of into this chase to isolate these resistance genes and try to understand how they conferred robust resistance. And for this work, I decided to work on rice for a number of reasons. So number one, it's a staple food for more than half the world's people. So if you can make small changes in the plant's ability to fight disease or tolerate environmental stress, you could potentially benefit the lives of millions and millions of farmers. And also, when I was starting my work at Cornell as a postdoctoral fellow, rice was emerging as a model system. And that means, for those of you that are not biologists, that it's easy to work with. It's genetically tractable. So I decided to study a bacterial blight of rice. So this is a bacterial pathogen that infects the leaves of rice and can cause very serious diseases. It can affect, farmers can lose half their crop to this bacterial infection, especially in Africa and Asia. And what you see here are leaves that have been infected with the pathogen, and the pathogen is actually oozing out of the leaves. So that's, it has a yellow pigment. So I spent some time trying to isolate this gene for resistance that was first discovered in Mali in a wild species called Oryzolonga staminata. So this is a weedy rice relative that farmers don't eat, but it is related and you can make genetic crosses. So scientists were able to show that this particular wild species conferred resistance to all strains of the pathogen. And a really wonderful colleague, Gurdev Kush, who was working at the International Rice Research Institute, made the initial genetic crosses and named this resistance XA21 for Xanthomonas 21. And Gurdev Kush is now an emeritus professor at UC Davis. And so when I first got interested in this, he gave me a bag of seed and basically said, go for it. So with my colleagues, we were able to isolate this resistance gene. And what you see here are leaves, 2 leaves on the top of a conventional rice variety. And we've inoculated these leaves with the bacteria. We just take scissors, we dip them in the bacterial suspension, we clip off the end of the leaves. We wait two weeks. And you can see that the bacteria form very, very long lesions on this leaf. Well, we knew we had isolated the XA21 gene when we were able to introduce our candidate gene into this conventional rice variety and show that the plants engineered with the XA21 gene were very, very resistant to the pathogen. So this was super exciting. And so what is the gene? Those of you interested in biology, it's a receptor kinase. And what receptors do, they have domains that stick outside the cell, the plant cell, and they can recognize, in this case, a microbial molecule, and then send that signal inside the plant, and it launches a resistance response. So it's kind of like an antenna that is perched on the plant cell looking for trouble. And we learned very quickly after that the toll receptor in flies, which confers resistance to fungal pathogen, also had a very similar structure. And then in 1998, the TLR4 receptor was isolated from mice and was also shown to have the same structure. So since that time, there have been many, many of these immune receptors identified. There's just a handful in animals, but there's hundreds in plants. So this continues to be a really important area of research. And in fact, these animal immune receptors are very important for the initial response to COVID viral infection. And that's been in the news lately. So we know now that, I'm not going to give you all the details, but we know these receptors recognize small microbial molecules, and these molecules have been isolated, and we know that the rice pathogen produces a small little peptide that's recognized by XA21. And so we know from this work that immune receptors in plants and animals are remarkably similar. And here you have the mouse looking at the rice plant, and they recognize these small microbial molecules. Now, after we isolated that gene, I, of course, was interested in the broader aspects of cultivation of rice globally. And one of my colleagues approached me to ask me if I'd be interested in looking at tolerance to environmental stress. And of course I was. So one environmental stress that's predicted to be increasingly important as the climate changes is flooding. So the Intergovernmental Panel on Climate Change predicts that floods will occur more frequently and be of longer duration as the climate changes. So what you see here are some kids walking through their rice paddy in Bangladesh after a flood. Now, rice grows well in standing water, but if the rice is completely covered with water, like you see in the center of this rice paddy, most rice varieties will die. And this is very challenging in places like Bangladesh, where many farmers rely on rice for 2/3 of their calorie and live on less than $3 a day. So 25% of the world's rice is grown in these areas, Eastern India, Bangladesh. And my colleague told me about an ancient rice variety that was highly tolerant of submergence that had been found in Eastern India. And in fact, it's been traced back to probably being grown by farmers there 2,000 years ago. But unfortunately, farmers don't grow it today because it has very poor yield and very poor flavor. So what breeders do is they try to introduce a trait of interest, in this case, flood tolerance, and try to introduce it into varieties that are locally adapted and favored by farmers in that area. And this is something that my colleague David McKill and his graduate student Kinong Xu began to do. And they were able to roughly map this gene so they knew approximately where it was located on the rice chromosome. And we decided to try to see if we could isolate this gene, because if you could isolate the gene, you could introduce it more carefully and precisely into varieties grown by farmers in India and Bangladesh. So we were successful, and this shows you an experiment from my lab, where on the left, you see a rice variety that has been grown for 2 weeks in water and then completely flooded. And then it's been allowed to try to grow again. So 2 weeks without floods. And the water's been removed. And you can see it doesn't do very well. In fact, most of these conventional varieties will die under those conditions. Now on the right is the same plant that we engineered with the sub1A gene for submergence tolerance. And much to our delight, you could see it was very robust and green, and it could survive 2 weeks of flooding. Now, this was just a laboratory experiment, and we really wanted to know, of course, how this would work in farmers' fields. So my colleague Dave McKill carried out a breeding process called marker-assisted breeding to introduce this gene into varieties grown by farmers. And I'm going to show you a time-lapse sequence that was filmed at the International Rice Research Institute in the Philippines. And so you could see how the new variety performs compared to the conventional variety. So on the left is the sub-1 variety, and on the right is the conventional variety. They both grow very well at first. You can see they're growing very well, but then there's a flood that's applied for 2 weeks. And then even after the flood is removed, you can see that the conventional variety, IR64, does not yield very much grain, whereas the sub-1 variety has about 3-fold more grain. And I really like to show this video because it shows the power of genetics, how a single gene can have a dramatic effect in making a crop more climate resilient. So Dave led a number of field trials, and certainly in the last 4 or 5 years, there's been really intense flooding in India and Bangladesh. And the whole team was able to visit in 2008, and we met the farmers. And this is just a field trial that the farmers carried out. And you could see that the Swarna sub-1 variety does very well in the field and has about a 60% yield advantage. And that's just because of this one trait. This project was really exciting to be part of. And as Olga mentioned, last year, 6 million farmers grew sub-1 rice. Now, one question that comes up a lot when we're talking about agriculture and genetics is who benefits from this kind of new varieties? And this sub-1 rice has been studied by a number of groups, including Kyle Emmerich and his colleagues who were at UC Berkeley. He's now at Tufts. And this is some data from his work. So what you're looking at is a region of Eastern India called Orissa. And those black dots are 128 villages where he did his experiment. And so what he did was provide the sub-1 variety, or farmers could choose to grow control varieties, and follow the farmers throughout the season. And in red are the low-lying areas, and the white are the low-lying areas, and the red are the mountains. And what is interesting is that these low-lying areas, obviously they're going to be more flood-prone, but they have been occupied by lower-class social groups for hundreds and hundreds of years. And in these low-lying flood-prone areas, often farmers will lose their entire crop. And what Kyle was able to show is that the sub-1 conferred 45% higher yield. And in fact, some farmers were able to harvest grain where they had not been able to harvest grain before. So this kind of study shows that sub-1 rice disproportionately benefits socially disadvantaged group. So the power of genetics and the power of seed is all a farmer needs is a single seed to benefit from genetic improvements. So it's a very appropriate technology for farmers in poor areas. Okay, I wanted to switch a little bit to talk about a different pest in Bangladesh. So what you see is an insect that has burrowed into an eggplant. And the black you see is frass. That's what comes out the back end of the insects. Now this insect is a big problem in Bangladesh for farmers, where eggplant is the most important vegetable. In the past, farmers have had to spray their crops sometimes several times a week with insecticides to control this pest. If they didn't control the pest, they wouldn't be able to harvest much eggplant at all. So there has been, and this is a big problem in less developed parts of the world, because often farmers and their families don't have proper protection. Kids are going out and spraying insecticides, and there's a high frequency of flooding, of poisoning. The World Health Organization estimates that 300,000 people die every year because of insecticide overuse or misuse. So this is a recognized problem, and so scientists at the International Bangladeshi Institute and Cornell University decided to try to tackle this problem using a genetic approach that's based on an organic farming technique. So organic farmers will purchase an insecticide called dipel, and they'll spray it on their crops to control against pests. Now, dipel consists of bacteria that are expressing a protein called Bt, and this Bt protein is very effective in controlling many different types of pests. And it's considered non-toxic. It's been used for about 50 years, and it does not harm humans or fish or many other animals. So it doesn't always work, especially in Bangladesh, where it's very expensive, it's hard to access, and if you spray it on the plant, it doesn't prevent the insect from getting inside the eggplant. So geneticists at Cornell in Bangladesh decided to take the gene encoding this enzyme, so they just snip it out of the bacterial genome and they insert it into the eggplant genome. And they have been trialing these eggplants for about 4 years now, and so now we can see how well this works. And it works very well. So farmers in Bangladesh have reported reductions in poisonings. They have about a 55% increase in yield, and it's been really quite successful About 20 to 25% of Bangladeshi farmers are now growing BT eggplant, and it's considered to be one of the most quickly adopted seed technologies in the world. Okay, and my last example is golden rice. Some of you may have heard of this. This was a project that was funded by the nonprofit Rockefeller Foundation. And so we know that 500,000 children go blind every year in much of the world because of lack of vitamin A, and half of those children die. So scientists decided to fortify rice with beta-carotenoids, which is a precursor of vitamin A. And normally in the rice grain, no vitamin A is produced. So in the engineered line that you can see here, it's very golden, hence it's called golden rice. And it has high levels of the precursor of vitamin A. This has been a really very, very exciting project, but also a controversial project. So even just a few years ago, there were people that were against modern biotechnology were pulling out these seedlings from test plots in the Philippines. And you know, from a scientific point of view and a humanitarian point of view, it's somewhat shocking, because you just wonder if the people pulling out the plants understand they're destroying not only a research experiment, but also potentially medicines that will save the lives of young children. So this has now been approved for consumption in many countries around the world, but it's still not in the hands of farmers because there's a lot of concern about modern genetic approaches. So that brings us to this last question. How do you know foods with new genes are safe to eat? And in a sense, this is a question I get asked a lot, and farmers get asked a lot. And really, what we can say is that genetic engineering, which is just one of many forms of genetic improvement, but genetic engineering, where you take a gene from one species and you put it in another species, has now been used for more than 40 years, since Mount St. Helens exploded. And it's been used in medicine, in wine, it's been used in plants, and there's not a single instant of harm to human health or the environment. And still, the other really interesting thing of our time, I am just going to come right out and say that, is that there's so much misinformation. It's really hard for consumers to know where to go to get accurate information. And, I don't want you to take my opinion, because opinions don't matter, and science is not a belief system. So we have to look at the evidence. And every scientific organization in the world has concluded that the genetically engineered crops currently on the market are safe to eat and safe for the environment, and that every crop has to be looked at on a case-by-case basis. And these are the same organizations that many of us turn to when we want information on the safety of vaccines or the reality of climate change. So I want to just finish with, so my husband's a farmer, he's an organic farmer, and we talk a lot about this. And we really think that instead of worrying about genes in our food, we need to have a more global, humanitarian, sustainable perspective, and really ask how we can enhance food security for the poorest people in the world, how we can minimize environmental degradation. We need to support farmers in rural communities and keep food affordable. And of course, we need to foster soil fertility and reduce harmful inputs. So I think that's really all I wanted to say today. Of course, this work, I've had a lot of good fortune working with many people over many years, and I guess that's it. So please let me know if you have questions, and I will stop the share if I can figure out how to do that.
Pam, I can stop it. Oh, you got it.
Yeah.
So I guess one question, Pam, this is Olga, that was sent in earlier, and I'm just rephrasing a little bit because you talked about, the importance of looking at the science to determine on if what we're eating is safe for our health. But do you know, and you said we need to look at case by case basis, do you know if there's been any studies since so many subsistence farmers have started growing the climate resistant rice? Are there any studies that have been conducted on the health of those people who are consuming that rice, specifically on their digestion or anything like that, specifically around that rice?
Well, no one's looked at the digestion of farmers eating that rice, but I think one way to think about it is it's a gene that has been consumed 2,000 years ago. It's been out of the modern varieties, and it's really been brought in from those ancient varieties. So it's not like something that comes from outer space or something like that. And we know a lot about that gene and what it does. And it's not involved in producing compounds that would affect a digestive system. So every plant that we eat has a completely different metabolic profile, and every variety has a different metabolic profile. And so, of course, it would be impossible to look at the effect of any of the foods we eat and do studies on digestive systems. And in fact, probably maybe gluten has been studied a lot, but most molecules have not been studied to that level.
Great, thanks, Pam. Another question from Jim. He says, thanks for an inspiring talk. With regard to keeping food affordable, who owns the patents for the sub-1 BT and golden rice, and how are they approaching distribution of these seeds? Following up to that, I had also asked you earlier to talk about the differences between how seeds are distributed in developing world versus United States. So those seem connected.
Okay, thanks for that question. So all the crops I mentioned, the sub-1 rice, the BT eggplant, golden rice, those are all in the public domain. So what that means that farmers don't pay anything more for the seed. And particularly, I'm most familiar, of course, with the sub-1 rice because my colleagues are the ones that are bulking that up and distributing it. So in the developed world, so United States and Europe, most farmers will buy their seed from seed companies, whether you're an organic farmer or a conventional farmer, because it's really a capitalist system here, right? So farmers buy the seed, they plant the seed, they sell the produce. But in the less developed world, it's almost completely different. So the farmers I'm talking about are generally subsistence farmers, so they only grow enough food to feed themselves and their family. So when you're living on less than $3 a day, you don't have, you're not able to afford to buy the seed. And so they generally get their seed from national breeding programs. So in India and Bangladesh, the Katak Rice Research Institute, it's a place we're able to visit. They have really talented breeders there that do the field tests, and then they distribute the seed from there. So the sub-1 rice goes through the exact same seed distribution system as any other seed that farmers would obtain. And so that's the same with golden rice in the Philippines, and it's the same for BT eggplant.
Excellent. And I think Eric was asking a similar question. Eric, if you have a different question, just throw it in the chat. Anne is asking if you could please discuss Monsanto and Roundup Ready crops with the consequent overuse of herbicides and the bullying of non-compliant farmers. Why is there such resistance to labeling GMOs so that people can avoid supporting companies like Monsanto?
Okay, so as I mentioned, in the United States, most farmers buy their seed from companies, and Monsanto doesn't exist anymore, but there are other big seed companies that farmers buy their seed from, including your favorite organic farmer down the street. They're buying their seeds from the same companies as other farmers. And so I think there's certainly a lot of discussion that could be had of whether we want big seed companies or not, but that is the case in the United States. And we do hope that that seeds are not sold unless they're safe, and that's why we have a very complex regulatory network that looks at these seeds for sale. Yeah, so one thing I like to make clear is, I actually don't use the term GMO because it means different things to different people. So some people think it means Monsanto. Some people think it means helping save the lives of children in the Philippines. And some people think it means patents. And it's just, and it's actually, the FDA doesn't use that term because everything we eat is genetically altered in some manner. And so it's really a confusing term. And so I think it's really good, and I like this questions because you're narrowing down on a specific question about seeds in general, but about a particular company, for example. But I don't think we can talk about GMOs as a group because you can't really compare golden rice, which has been developed by a nonprofit group to save the lives of young children, with maybe canola in Canada, which is for-profit farmers that are extracting oils and bottling canola oil. These are just so different. I think sometimes the questions reflect that there's not always a super great understanding of what farmers are doing and what their challenges are. And yeah, okay, so the other part of that question was herbicide tolerant crops, and this is a crop I did not talk about. And so it's besides BT, so BT crops are some of the most important widely distributed, well, BT crops are probably the most widely distributed genetically engineered crop in the world. So that's insect resistance. So the farmers have massively reduced their insecticide sprays. There's another crop that the questioner asked a question about is called herbicide tolerant crops. And those are more controversial because instead of removing pesticide sprays, the farmers are shifting to a different type of herbicide. And so, yeah, thank you for that question. And I'm happy to take other questions.
Pam, instead of saying GMO, someone is asking, what's the correct term? How could we speak more specifically about genetically manipulated?
I think it's really useful to say something like a submergence or flood tolerant rice, or a insect resistant eggplant, or an herbicide tolerant canola. So you're talking specifically about that crop because there's very interesting questions about every crop we eat, right? But they're different for every crop. So you can't really generalize about the whether genetic technology is good or bad because it's just a technology which is neutral. So what matters is to ask these bigger questions, is how does that particular seed enhance sustainability of a particular farming system in a particular location?
That makes sense. A follow-up question about the rice varieties. Are consumers accepting this quote unquote strange looking rice? So I guess this is about the golden rice.
Oh, the golden rice. Well, that's a really good question. And it's not out yet. I mean, it's been almost my entire career that this has been a very interesting project, and it's still not out. So there's still a lot of sort of anti-biotechnology activism. It's a nonprofit group, the International Rice Research Institute, that was developing the rice. So it goes really slowly. They don't have the resources, for example, as a big company like Monsanto. They do have the rice that's been field tested in the Philippines and in Bangladesh. And they've also doing a lot of studies on whether or not farmers and their families will be able to be comfortable eating it. And this is where you get to a lot of socioeconomic questions. So if a mother or father knows that it may save the life of their children if the child is malnourished, I mean, I think all of us can understand, well, of course, you would want that rice. But if they get misinformation or disinformation about that crop. And sadly, in our world today, there's a lot of disinformation. And even with golden rice, you hear things like, it's going to give you cancer. And if somebody hears that, and it doesn't matter if they're educated or not educated, it frightens the person. And so really, I think we don't know the answer to that, how well it's going, how well the uptake will go. And it takes a while before parents see that the child's health is improving, and it takes a while for that information to spread through informal networks. So it's really a matter of trust in our scientific institutions, which, just to get a little political here, that we have seen a lot of distrust sowed by politicians. And I think that's one of the things I care most about this time in my life, is really building back trust in our scientific institutions and trying to deal with this massive disinformation we have in many aspects of our lives.
So Pam, an appropriate follow-up to that was, someone is asking about companies like Monsanto or Bayer, or some of these big companies, and how they've been allowed to influence the regulatory system. This person asks how eroded trust in this is. Trust has been eroded. Do you have any thoughts about how to restore the trust so truly beneficial and safe crops aren't tarnished?
Yeah, I think that there's absolutely, we can all think of examples in our scientific institutions, our regulatory institutions, or our insurance companies, or pharmacy companies, where there's been really bad examples, right? Of bad things have been happened, and that then we can conclude these companies don't deserve our trust. So, I think it comes back to rebuilding our government, right? So we can trust our government to regulate the regulators. We can trust our corporations to pay their taxes, to support research. And I think these are just fundamentally important in questions. And, but I would say, at least the crops that I'm most involved with, which is this, the sub-1 rice, there have, as far as I know, not been any regulatory snafus. In fact, because it's developed through micro-assisted breeding, it's not even regulated at all. So most of the foods that you eat are not regulated at all. So the only foods that are regulated pretty much right now are those developed through genetic engineering. For example, putting that Bt gene into the crop. And for the Bt crops, there was a pretty nice rollout of those crops. Farmers were mandated to have an integrated pest management system. And I think most agricultural scientists would say that the herbicide tolerant rollout was not good, because farmers were not encouraged to use an integrated weed management. Often farmers would just spray the same herbicide over and over. And we know that's not a sustainable practice.
Pam, following up on the Bt line, someone is asking, how are the consequences of Bt, or what are the consequences of the Bt genetics on insect life? Insects, butterflies are in decline. Where is the balance between saving crops and saving species diversity? And maybe are you saying that people are overspraying and that's one of the reasons why we've seen the decline, or is there not a connection there?
So there's not a connection. I was talking about the herbicide. So people are spraying herbicides somewhat indiscriminately, but the Bt crops have been really robust, and I think much data showing they're very healthy for ecosystems. The reason is they're very specific to target pests, so they do not kill butterflies. And there's been studies, so that's why organic farmers love it. Organic farmers have been spraying Bt for years. And so there's been really interesting studies to show that insect diversity is coming back into fields where Bt sprays or Bt crops are used, because instead of spraying a broad spectrum insecticide that kills everything, you're producing a compound that targets only those pests. And so there's been a lot of interesting studies about that.
There's a couple questions here getting into the specifics around rice. How much genetic variability is there in golden rice For example, if the temperatures or the amount of rainfall change, are these homogeneous, or can some survive when other plants perish?
Okay, so that's interesting. If you combine the submergence tolerant trait with the golden rice trait, you would have vitamin A enriched rice that survives flooding. And the reason I'm giving you that example is to sort of convey that every trait has a different effect. So the golden rice traits are for nutrition. So you're producing the compound that we eat in carrots, right? And those compounds do not affect flood tolerance or drought tolerance. Whereas the gene that we engineered into the flood tolerant rice, that gene has no effect on nutrition. It doesn't change the nutritional profile, but it does allow the plant to survive floods. So that's what's so interesting and fun about plant genetics, because every gene does something different. And what breeders do, and they've done, ever since the beginning of breeding, is they've been combining different genes together. So what we eat today is nothing like our ancestors ate 10,000 years ago, because we've had 10,000 years of breeding. And essentially what breeding is bringing in beneficial traits that are either benefiting the farmer, they grow better, or maybe they're benefiting the nutrition or flavor or color. And so there's a lot of interesting breeding strategies.
Pam, following up to that, how do plant breeding strategies or practices taking place today differ in the way that we were manipulating genes maybe through crossbreeding that have been done thousands of years ago? Can you get into the specifics of what those different techniques are and how it's evolved?
Sure. I have a nice slide, but I didn't show it. So 10,000 years ago, it was this thing called primitive domestication. So our ancestors would plant some seeds, and then they would harvest some seed and replant it. So that kind of selection starts to change the genetic makeup, because pretty soon you don't have as a wide a diversity. So that's kind of considered primitive domestication. And then with the discoveries of Mendel, we had a better understanding of genetics and hybridization. And so farmers started using cross-pollination. They would sometimes cross-pollinate two different species together and come up with a new variety that didn't even exist in nature. And actually in the, 1850s, when hybrids started to become very important, both for horticulture and agriculture, there were the same kind of discussions then as there are today, that, this is not God's will to make these new varieties. And there was an article in Scientific American from back then, and it was really remarkable how similar those kinds of fears and concerns were. And it's true, it's different. Nothing we eat today, virtually nothing we eat today is found in nature, except, here on the West Coast, we have Pacific salmon. On the East Coast, you can hunt for blueberries, we can hunt for mushrooms, but most of the food we eat does not occur in nature at all. And so there has been this change. And then more recently, genetic engineering, like I said, is 40 years old. It's not even a new technology anymore, but it's still, there's still a lot of news about it. And, I guess I have to say a lot of marketing about it, because if you write non-GMO on your label, you can sell the food for twice as much or something. So it's not really based on safety or sustainability. It's really more surrounding marketing. I think why that sort of concern has lasted for so long. But now we also have new technologies such as genome editing, which is a technology where you can change just a few base pairs in a gene. So you can make very precise edits. And this was actually, since 2012, it's just taken the world of genetics by a storm. You can do genome editing in virtually any organism. And it's very precise, and it's certainly not something we could do even 15 years ago. It couldn't have been done in plants. So there has been just a huge change over time. And that's why I really think it's important to think about the challenges faced by farmers and to try to enhance the sustainability of our farms because those are the issues that matter. The genetic technologies will just keep changing, and it's almost impossible for your average consumer to keep up with those genetic technologies. Even me, I have to work pretty hard to catch up with my colleagues. So I really think we need to think about what that particular seed is going to do in that particular environment for that farmer. How is it going to reduce inputs, and to think about those other aspects that I talked about.
So Pam, we have about 5 minutes left. I've got 2 questions I'll ask, and then I'll just seed this by saying if anyone wants to stay a little bit later and have a little bit more informal just dialogue with Pam, we'll be open to that. So there is time for that. One question was, what was the miracle rice in the Philippines in the 1960s? What was that all about?
Oh yeah, that's a really good question. So miracle rice was IR8, International Rice Research Institute, line 8. It was called miracle rice because it was a genetic change that instead of the plant growing tall and then during storms, it would fall into the mud and you'd lose your grain. So miracle rice was a dwarf rice that allowed the farmer to harvest higher yield simply because it was more compact. So that was a genetic change. And that gene has now been isolated, and we know what it is.
I don't think there's a question that you can't answer, Pam. But here's a good one. And this is from our friend Mateo. Any thoughts on why some people or groups that advocate for believing the science, especially nowadays with COVID, or about something like climate change, are suspicious of other science, in particular, genetically modified conversations, technology?
Well, that's a good question. And, sometimes it's a matter of, your milieu, where you're living, what you're buying, what Facebook you see, what Facebook you're reading. And I think it's really an important question because, and it's a failure of scientists as a scientific community. So why is it that we have failed? So I would say that here on the West Coast, not all of you are on the West Coast, but many people support the science of climate change. But they might, for example, have heard that modern genetics is unsafe. And so it's really sometimes what you hear from your friends and family. And I think we have failed to really explain that when you're looking at evidence that you need to go to, and none of us understand all these scientific questions. So we need to have some sort of, again, trust in our institutions. And I think people that support the science that the climate is changing might not always recognize that there are scientific institutions that are making those decisions. So they hear, well, the scientists say that, but they're not really maybe discerning that it's the National Academy of Sciences that has done studies for many, many years that has made that decision, or I shouldn't say decision, but that consensus. And it's the same for genetics. These are not different scientific organizations. They're the same organizations that have concluded that the crops on the market are safe to eat, the climate is changing, and that you need your flu shot. These are the same scientific organizations. And it's not like a scientist in the United States is different than a scientist in Europe. So the scientists globally generally have the same consensus, but it's the political systems that will take a different approach.
Pam, that's excellent. And there's some people writing into the chat just saying how much they appreciate this talk and how informative it's been. I'll ask you, and I can follow up with everyone after with an email, but if you have specific resources that would be helpful for those of us who want to become more informed here, as opposed to just following, the tweets of our cousins who are talking about GMO, this and that, I would really appreciate that. I'd be happy to share that with everyone who attended the talk. Is there anything off the top of your head that you think we should be tuning into now?
Okay, well, on my website, I have a bunch of resources to National Academy of Science, World Health Organization, USDA. Yeah, you can also check out our book that I wrote with my husband, an organic farmer. We have loads of citations. My son just told me the other day, like, why so many citations? Because he's finally getting around to reading it. So I'd say if you go to my website, you can just Google Pamela Ronald, UC Davis, it'll probably come up. And yeah, I'm happy to share that.
Okay, that is excellent. And I'm keeping track of the time. So I'm going to jump in. And again, if anyone would like to stay and talk to Pam a little bit longer, feel free to just stick around. But I appreciate having those resources and also you mentioning your book. And I'll put all of that together in an email and send it out to everyone most likely tomorrow. So I wanted to thank Pam for giving your time so generously to make this talk possible. I can say on behalf of this group that I've learned a lot, and I have much to share with the people who I live with around the kitchen table, since those are the only people that I see nowadays. I know that Pam will be here a little bit longer and is looking forward to following up with fellow Reedies. So I'm just going to wrap up with a little bit of informal or a few last minute details. I would love to get your feedback on this talk. I manage all of these Reed Remote talks, so I'm actually going to throw a survey into the chat, which you'll also receive via email tomorrow. If you would like to share this talk with others, this is a great way to spread facts out to the community. Feel free to share it. We will upload this to the Reed Remote webpage, and you'll be able to access that. I will send you those links in my follow-up email. And on our Reed Remote webpage, you'll also see our upcoming events like our Forum for the Advancing Reed, which this year is taking place online. And we'll be hosting a talk by President Bilger, where you can hear firsthand what it's like to manage a college during the pandemic. It's been wild. We'll also have a faculty talk by Professor Paul Gronke, who is a distinguished professor of political science here. He's going to be talking about elections. Sorry to even say that word on this talk, but we are going to be spreading facts about voting during this Forum for Advancing Reed. And then we'll also be showcasing over 40 students who are reporting back about their experience with remote summer internships. So it's going to be a fascinating weekend. It's taking place between August 16th and 18th. We've tried to spread it out over multiple days, and you're all invited. I will follow up with that in my email as well. I wish you all well. Thank you so much for attending. We love to stay engaged with Reedies. I work for alumni programs. I am here for you. If there's anything we can do for you during this very challenging time, please don't be a stranger and reach out directly. For those of you who are wanting to stay a little bit later and chat with Pam, just stick around. And to everyone else, I wish you good night. Thanks for joining us.
Thanks so much, Olga. And I look forward to talking to my old buddies that I see your names, but I do not yet see your faces. So please hang out.
Hang on.